All right, so as it relates to the shape of bacteria, we can use the shape of these bacteria to aid in diagnosis. And one thing I want you to note is that the shape of bacteria, they come in different, it varies, right? There's a variation in the shapes. So these are the most common shape of bacteria that causes human disease, right? Most human pathogen, they fall under, or they have these shape here. All right, so the first one we're gonna look at are those cocci. All right, those cocci. So both streptococcus and staphylococcus, it appear cocci in shape, right? It appear as a circle, right? But the arrangement is different, right? The arrangement is different. Streptococcus, as the name suggests, it occurs in chains. So it's cocci, and it appears as cocci in chains, right? Cocci in chains. And streptococcus appear in clusters, right? Streptococcus appear in clusters, right? And it get like it look like it look like a grape-like appearance. It got like a grape-like appearance after gram stain. After you would have gram stained and then visualize it under the microscope. All right. So for bacillus, so streptobacilli also occur in chains, but yeah, streptobacilli occur in chains also, or gram negative those gram negative organisms such as members of the Enterobacteriaceae family like E. coli, Klebsiella, Proteus. They also appear bacillus. They appear like a rod, right? Rod shape. Then we have Coccobacilli, a good example of Coccobacilli. So Coccobacilli is this here, and this Coccobacilli is a variant between the cocci and the bacilli. It's not as small as a bacilli, but it's not as long as a it's not as small as a cocci, but it's not as long as a bacilli. It's like just in between. And a good example is lactobacilli. L A C. Well, lactobacilli. All right. Then we also have Vibrio. Right. Vibrio is this here. Vibrio, and a good example of Vibrio shape is Vibrio cholerae that we would have talked about in the last class. Then we have Bacillus, or you see rod shape or bullet shape organism. Those are, like as I mentioned before, members of the Enterobacteriaceae family like E. coli, Klebsiella. And we have the Spirillum and spirochetes, right? So these are things that we already would have went through. Bacteria prokaryotic cells, they don't have nucleus. No, they don't have a well-defined nucleus, or the nucleus is not membrane bound, and also they don't have membrane bound organelles. So their DNA they have they're single circular chromosomal DNA, and these chromosomal DNA is found in the nucleolus. They also have DNA found within the plasmid, right? DNA found within the plasmid, and this DNA is known as extra chromosomal DNA, right? That's known as extra chromosomal DNA, and this DNA can be transferred from one organism to the next via conjugation, right? This DNA can be transferred from one organism to the next via conjugation, and it can be transferred. And the structure that is transferred via is the pili, right? The pili. So I don't know if you can remember the function of histones. So histones are important for packing DNA, right? Packing DNA, and to make it make the DNA well, yeah, to pack it and to make to create more space so that more DNA can be fit. But the thing is, bacteria don't have a lot of DNA, so there's no need for histone. Our chromosome contains histone because I don't know if you would have heard, if you uncoil our chromosome or uncoil our DNA, it can go around the earth 2 times and back. Right? So the cell wall of bacteria contains a very important structure known as peptidoglycan. Right? Peptidoglycan. Right? And it contains two polymer, NAG, NAG, and acetylmuramic acid, and NAG and glutamate. All right. So it also contains small ribosomal units, and these ribosomal units are very important for the production of protein or production of protein synthesis. And of course, they are produced via binary fission, right? This is very important to remember, right? They are produced via binary fission. So these are the structures again that we're gonna be talking about. I think we spoke about a few earlier, but we're gonna get into a little bit details with these structures and their function. So one of the things that I want you to note is that some of these structures that are present in this bacterial cell form something known as virulence factor or virulent factor, right? Virulent factor. So virulent factors are those structures or components of a bacterial cell that increases its probability of causing a disease. And by increasing the probability, I mean it. These virulent factors are important for things like attachment, important important for things like evading the host immune response, and then setting up an infection. Right. So let's get into these bacterial structures. So the first thing that we're going to be looking at is the cytoplasm. So let me go back. So the cytoplasm here is this bluish, the entire bluish component of the cell. And as you can see, within the bluish component, there are structures diffused within the cytoplasm, within within the cytoplasm. So the cytoplasm is also known as protoplasm. So these two words are used interchangeably, and it is this jelly-like matrix, right? Is a jelly-like matrix here, right? This gel-like matrix, and it is the site where metabolic reaction occur, right? Metabolic reaction occur, both anabolic and catabolic reactions. That plays an integral role for the cell survival, right? As you can see, there are enzymes, and enzyme plays an important role in metabolism, right? Both anabolic and catabolic, right? This is the site where nutrients are stored, where waste product, you can also find waste product gases. It also has a number of structures diffused within the cytoplasm. So the cytoplasm is a site of growth, site of metabolism, and site of replication. And that would make sense because because of those enzymes that are present. So one of the things that I also want you guys to note is that that bacterial cell, they store food when food becomes abundant, right? They store food when food becomes abundant, and they also store their food in the cytoplasm, right? They store the food within the cytoplasm, and they store food in the form of granules or inclusion bodies. right? They store the food in the form of granules or inclusion body. So one of the things that that I want you to note is that these granules and inclusion body, they can sometimes they can be used, sometimes they can have um, diagnostic properties, right? Because, for example, in Corynebacterium diphtheria, they store their food when protein become excessive, and they store it in the form of something known as volutin. VOLUTIN, right, in the form of volutin. And we can stay in the volutin and it and and identify the organism, right? Identify the organism. All right. So another important component of the bacterial cell is the ribosome, right? Is the ribosome, and I'm certain by now all of you know the function of the ribosome, right? All of you know the function of the ribosome. So